Next topic is replication. As you know, the term replica means copy. So replication is of mainly two types. One is normal replication and one is abnormal or defective replication. This defective replication is also known as DI, defective interference particle, or von Magnus phenomenon. And the example here is the influenza viruses. Influenza viruses. So we'll discuss this replication, that is copy of viruses, in two modes. One is normal, one is defective. So as you know, the replication means making copy. If we compare bacteria and viruses, bacteria, this is the E. coli, this is the E. coli. It multiplies in 20 minutes. One bacteria multiplies into 2 and this 2 to 4. 2 to 4. So this is how the bacteria multiply, bacteria replicate. But viruses are a little bit different. One virus enters and it multiply within the nucleus or cytoplasm, and for example 150 comes out. So the replication or the multiplication of virus is little bit different from the bacteria. The steps involved in virus, they are different, and steps involved in bacterial multiplication or growth, they are different. So we'll see how the bacteria divide and how these viruses multiply from their growth curve. Here we'll see bacterial growth curve versus viral growth curve which is also known as one step growth curve. This was given by D and E, that is Delbruck and Ellis. This was given by Delbruck and Ellis. In 1939, they worked on E. coli bacteria and T4 bacteriophages. So, this is the curve, bacterial growth curve, which is nothing but S-shaped growth curve, and here is the viral curve. Here is the viral curve. So in bacterial growth curve, there are 4 steps, 1, 2, 3 and 4. Similarly, here also 1, 2, 3 and 4. This phase is known as lag phase, log phase, stationary, and decline, phase of decline. So this is how the bacteria growth curve is look like: S-shaped, lag, log, stationary, and decline. But viral growth curve, one-step growth curve, explained by Delbruck and Ellis on E. coli with the T4 bacteriophage, which is a virus which attack the bacteria. Here are four phases. Here are similar to this. There are four phases. This number one is adsorption and penetration or entry. In this part there is adsorption and entry. Number two, this phase is known as eclipse period, EP, eclipse period, which involve uncoating and replication. Number three, this is the number three phase. So what steps are there in number three? Assembly of viruses and assembly, and this is maturation, not release. This is maturation, assembly and maturation. Number three. Then number four is the burst phase, which is known as the release of viruses from the cell to the outside. So this is the one-step growth curve having four particular steps or phases. Number one is adsorption and entry. Two is uncoating and replication, and this two is known as eclipse phase. Eclipse means to hide or to disappear. So in this phase, inside the cell, we cannot see the virus particle. Then number three is the assembly and maturation, and four is the release. And this 2 plus 3, this one till the release, this is known as latent period or latent phase, LP. So this number 2 and 3, we call it latent phase. So latent phase means beginning of the eclipse and until the release of the virus outside of the cell. So this is the viral growth curve, one-step growth curve. Similar to bacterial, there are four phases. Number one is the attachment and the entry of the virus. Number two is the eclipse period. Number three is the assembly and maturation. Four is the burst phase or the release or exit phase of the virus from the cell. And this eclipse period is the period where steps like this uncoating, replication, transcription, and translation takes place. So eclipse word itself indicates hide or disappearance of the virus particle. Then 2 and 3 combinedly known as latent phase of the virus, which include eclipse plus these two, assembly and maturation. Next, we'll discuss the replication rule of 3. Rule of 3. So, Rule of 3 means there are total 9 steps which are divided into 3 parts. First 3 are APU. APU, these are the 3 steps, and last 3 are AMR. AMR steps. And in between here central part is in the center we have central dogma. We have central dogma of the biology. And in central dogma there are 3 steps again: transcription, translation and replication. So central dogma is nothing but DNA to RNA to protein, where this is replication, transcription and translation. So this is known as the rule of 3. 3 steps here, 3 in the center and 3 at the end. So total 9 steps in replication. These are known as: A means attachment or adsorption. Attachment or adsorption. P means penetration, and this U is the uncoating. So these are the 3 steps. Then central dogma 3 steps are replication, then transcription. Transcription is of 2 types: early transcription and late transcription. Then similarly we have translation. Translation is also early translation and late translation. So we have 3 steps here, and at the end these 3 steps are assembly, and M is the maturation, and R is the release. So this is how the rule of 3 works, with the attachment of the virus with the host cell, and last is the R, release of the virus from the cell. So first we'll discuss these 3 steps: attachment, penetration, uncoating. So rule of 3, that is your APU and central dogma and EMR. So first is the attachment. So for the attachment or adsorption, over this cell we have cell membrane. This is the cell membrane. Cell membrane has a receptor and virus has this ligand. Virus is having ligand. Suppose this is the influenza A virus, which is having ligand is the heme agglutinin and receptor is the salic acid, which is also known as N-acetyl neuraminic acid. So receptor and ligand. This step is known as the attachment of the ligand with the receptor. Host cell receptor. In case of influenza viruses, we have different types of receptors. This sialic acid this is the cell membrane, and these are the some protein parts which is having, these are the carbohydrate, and the last carbohydrate, this is known as galactose residue. Galactose residue. And this is your sialic acid. This last one is. Based on this linkage, there are three types of sialic acid receptors. There are three types of receptors. Number one, avian receptors. In the gut epithelium, of ducks, we have alpha 2,3 sialic acid receptor. Alpha 2,3 sialic acid receptor. Then, in case of human being, upper respiratory tract is having alpha 2,6 sialic acid receptor. Lower respiratory tract is having alpha 2,3 receptors. These upper respiratory tract receptors are predominant. These are the predominant, more in number, and they are less in number. This alpha 2,3. Then in case of pigs, they have both alpha 2,3 and alpha 2,6. So in case of swine or pig, these receptor, avian or human, they mix here. This is the avian, this is human. So both receptor mix in case of pigs, so they are known as mixing vessels for avian influenza or influenza type A viruses. Mixing vessels are the pig. So birds contain alpha 2,3 receptor at this place. Suppose this cell is gut epithelium in case of birds, and this in case of human, this is upper respiratory tract epithelium. Then the nature of receptor will be, the receptors will be this one, alpha 2,6 sialic acid. So this cell depend on which species, avian, human or swine, and which organ, gut epithelium or respiratory epithelium. These sialic acid receptors are different, and this difference is because of the linkages between sialic acid and galactose residue. This linkage. So this linkage in avian birds is alpha 2,3 linkage. In human, both types of linkage, but predominant or maximum abundance linkage is in upper respiratory tract and less or few in lower respiratory tract. Pigs are the species which contain both type of receptor. So they are known as mixing vessels for the avian influenza. They mix these type of receptors and are responsible for recombination of a new variant. So this was all about the different types of sialic acid receptors in different species. These receptors are different, but the binding ligand for all these receptors is the same. That is the heme agglutinin, HA, which is a spike, which is a glycoprotein on the surface of the virus. So similarly, uh, this attachment receptor and ligand will discuss few more examples from other viruses. So this was from influenza viruses. Influenza type A viruses. Then here is the virus and here is the receptors. So first interaction of virus with receptor is the steps attachment. So number one is the parvovirus. Parvovirus. And receptor is known as transferrin receptor. Transferrin receptor. Then influenza type A Newcastle disease, that is sialic acid. Then number 3, influenza virus type C. Type C is having different receptor, that is known as 9O acetyl sialic acid. Then number 4, rabies. Rabies is having N-acetylcholine, means cholinergic receptor, and one more is the NCAM, some adhesion molecules. Number 5 is pseudorabies. Pseudorabies. In case of pseudorabies, the receptor is belong to CD155. CD155. Number 6, canine distemper. Canine distemper. Receptor is known as SLAM, that is more predominant, and in epithelial cell it is nectin-4. For epithelial cell it is nectin-4. And one more is simian virus SV40. The receptor is known as major histocompatibility MSC type 1. MSC type 1. Then the last one is the FMD virus. Last one is the FMD virus. Foot and mouth disease, FMD virus. FMD, if it is wild type or if it is cell culture adapted. For FMD virus, if it is wild type, then the receptors is integrins. Receptor is known as integrin. And if it is cell culture adapted, then the receptor is heparin sulfate. Heparin sulfate. If this is not given that FMD virus have wild type or cell culture, then the answer will go to integrins. If it is specified the virus is cell culture adapted, then it will be heparin sulfate. So this was about the attachment step. Then comes to rule of 3. After attachment, P is the penetration step. P is known as the penetration or entry or uptake. These all are synonyms. So penetration. Number 1, some viruses are non-enveloped. We call them naked virus. Naked. And some viruses are enveloped. Some are enveloped virus. For these non-enveloped or naked virus, method is known pore mediated, that is via some channels. This in the cell membrane, some pores are created. So which virus enter through these pores after attachment? That is the, for example, poliovirus, which is a member of picoRNA, picornaviruses. Number two, perforations. Number two, perforate. Perforation, that is parvoviruses, canine parvovirus, through the perforation. Then these are the enveloped virus. Number one, fusion. Number two, endocytosis. Endocytosis. This endocytosis is mostly clathrin mediated endocytosis. This is the membrane where receptor and virus attachment is there. Then these are the clathrin proteins below the vesicle, and it enters the virus enter inside. These are the 8 segment. For example, this is the influenza virus having 8 segment, and this structure is known as endosome, and this is our virus. This is the virus particle. So endocytosis or direct fusion. There are some viruses which undergo direct fusion. They are the paramyxoviruses. For example, Ranikhet or Newcastle disease. So this is how the virus penetrate or they enter inside the cytoplasm. Then we'll go to the next step, that is the uncoating. Once it is inside, then there will be a release of this RNA or DNA from the will take uncoating of. Again, the example will be from the influenza virus, influenza A virus. So first it was salicylic acid type 2, 3 or 2, 6 or both, and then virus attaches here, this interaction. Then what happened? Clathrin mediated endocytosis. Clathrin mediated endocytosis. Then it comes inside. With like this. This structure is endosome. Inside the cell we have lysosome. This lysosome fuses with this structure. Then there are 8 segments, and then we have M2 channel. So in influenza viruses, uncoating step is facilitated. The main role of M2 ion channel. These which ion? They are proton ion channel. So once the lysosomal content is mixed with this, these proton channels allow the lysosomal proton influx. H plus. So protons comes inside this virus core, and because of the acidification, this uncoating is a pH dependent step, pH dependent event. So the pH of lysosome, H plus content, they enter into the virus through the M2 ion channel, proton channel, and make the interior of the virus very acidic, very acidic. So because of this, what comes out, all the 8 segment RNA, they comes out. So this is known as uncoating of the virus. So uncoating is facilitated by the, or main role played by the M2 ion channel, and it is pH dependent event. So pH provided by the lysosome, and H ions enter into the interior of the virus, and disruption occur, and RNA comes out. This RNA will enter further into the central dogma, that is into the replication, transcription and translation. So this is the uncoating of the influenza virus. So to prevent this uncoating, we have some drugs. We have some drugs which are known as, if we block this channel, we have M2 blocker. We have M2 blocker, and the drug is known as amantadine. So this drug can block the M2 channel, and we can block uncoating steps of the influenza virus. Once the uncoating is blocked, then we can say replication is blocked. Replication is blocked. So M2 blockers, they block the uncoating step. The example is amantadine. So today we discuss rule of 3 with the first initial 3 steps: attachment, penetration, uncoating. In the next section we'll discuss central dogma, and this attachment we took example of the influenza viruses. These hairs may be hemagglutinin, Na and M2 ion channel, and this was the salicylic acid. This was the attachment step. Then this was the penetration step, that was clathrin mediated endocytosis. It becomes endosome. Then lysosome fuses. Then what comes, the steps of uncoating. This is the lysosome, which is filled with protons, and M2 channels allow this proton enter inside, and what comes out, the 8 segmented genome, that is RNA, which is single stranded negative sense. So these are the 3 steps from the rule of 3. In next we'll discuss the central dogma, where these segmented 8 segments, they are going to enter into central dogma, that is transcription, translation and replication.